The law of independent assortment deals with genes that are on non-homologous chromosomes, so the long yellow chromosome versus the short blue chromosome. Again, we're starting with the P0 parental generation, that is homozygous, true breeding, this time both for the A trait on the long chromosome and the B trait on the short chromosomes. I've skipped ahead to the F1 generation. These offspring are heterozygous at the A locus and at the B locus. Now this diploid cell, which is 2N, is ready to prepare for meiosis. First, it does DNA replication during S phase of interphase. Now every chromosome is made of 2 chromatids. There are still only 4 chromosomes present, 2 long and 2 short. This cell can now go through meiosis 1 and separate homologous chromosomes. When the chromosomes are lined up in this orientation, both the little a allele and the big A allele go into one cell, and the big A allele and the big B allele go into another cell. Now, at the end of meiosis 2, we have 4 gametes, 2 that have the red and green chromosomes, little a and little b, and 2 that have the yellow and blue chromosomes, big A and big B. But these are not the only possible gametes we could get from meiosis in this organism. Instead, the chromosomes could have lined up like this at metaphase of meiosis 1, with big A and little b on the right, and little a and big B on the left. At the end of meiosis 1, we get one cell that has little a with big B, and the other cell that has big A now with little b. So at the end of meiosis 2, we end up with 2 different kinds of gametes. One that has big A with little b, and one that has little a with big B. So depending on how the chromosomes line up at metaphase of meiosis 1, we can get 4 different kinds of gametes from the same parent. We could get big A with big A and little a with little b, or we could get big A with little b or little a with big B. We can do a big Punnett square with this to figure out all the different genotypic combinations from parents of these genotypes, but it's actually easier to do math than to draw a big Punnett square. So look at the next video on how to use some math to determine the probability of a genotype or phenotype when following 2 or more genes at the same time.